Oh, the studies of twins here, as I just mentioned. You know, if you want to study things that are related to environmental factors, usually study non-identical twins. If they're expressing a particular trait, you can say, well, they're just, you know, brothers, or, you know, brother and sister. You can have non-identical brother and sister twins. So you can see that the trait is based on the environment. It's caused by environmental factors. And then if they are identical twins, and they are showing a specific trait, you say, well, that must be genetically, you know, it could be environmental, but could be genetic as well. There's a whole sets of twin studies that they come up with these correlations of how likely a trait is to be inherited and expressed due to the genes, the genetic composition. And you see here in case of this is asthma, studies that were done with asthma patients, when they studied monozygotic twins coming from the same egg, right? Those are the identical twins. They see a lot more correlation of these kids that would develop asthma, like 65% of these kids, that if one twin has asthma, the other one will have asthma as well. Now, if you see in case of dizygotic twins, they are just, you know, brother and sister, could be, you know, don't have to be twins. You'll see that the correlation is about 37%, which means anybody else in the gene population maybe, okay? Because the index of asthma is quite high these days. I'll just put this picture here. I think it's very cute. Two identical twins. My friend's daughter. She thought she was going to have one boy, and she ended up with two girls. And that's my boy, my brother's wedding. In the middle, like, what do I do here? You know? Yeah. But they're very cute and totally different personalities. I mean, you talk about epigenetics. Okay, different methylations on some chromosomes and different expressions. So here, in the case of studies of twins, just a little concept check. You can see if it has 100% concordance with both monozygotic and dizygotic. So here you're looking at a trait that shows up with both types of twins. Okay, so what can you say about what is controlling that trait, the expression of the trait? You can say, well, it could be genetically inherited, or it could be due to the environment. So in this case, if both sets of twins are showing the same trait, you can say both of genetic and environmental factors are having a big say on the expression of those genes. Okay, now the book is mentioning, you know, a little bit about when do you seek genetic counseling. Who will really go and seek genetic counseling? I think the most often what we see is parents who are ready to have a child, or a woman that is pregnant and she knows that there is a history of some type of either birth defect or some type of disease in the family or syndrome, and she wants to know what are the chances, or we have to do some genetic testing to know if this child is okay or not. Another reason that you can seek genetic counseling is also when you have a woman that is older, they say usually older than 35 years old, and she's going to get pregnant. There are chances that something's wrong with the egg, or there are some chances something's going to be wrong, you know, with the pregnancy, with the baby. So this is the type of person that goes to look for genetic counseling. And also when you have prenatal tests that are pre-genetic counseling, for example, when you have a woman that is relatively young, in the 20s, and it so happens that during urine tests a high level of alpha-ketoprotein showed up, and it's not supposed to be high. So then usually the OB is going to direct this person, look, let's go do further testing because maybe something is wrong with the baby. Okay, and then it happens sometimes right in the first month, third month of pregnancy, this level was so high and started to cause high blood pressure, preeclampsia. With an ultrasound to guide it, as we're going to see here. So let's go down the line. What do? Okay, you look at the baby, right? So prenatal tests. It is part of the chapter. Look at the ultrasound, and you can see now all the technology today. You can get these 3D ultrasounds, and the baby's moving all around and dancing and kicking. Your bladder, not a lot of fun, you know. But yeah, it's a lot of fun to watch the baby. Oh, look, oh, that's the nose, that's the leg, you know. Okay. We did amniocentesis, insert this nice, delicate looking needle. So the needle has to be, you know, very scary. You guide it with the ultrasound. So what this exam is doing is catching up some of that amniotic fluid that is present here. Because in this amniotic fluid, you have a lot of cells that are coming from the baby, not only the cells from the mother. You have a lot of cells coming up from the baby. So once they collect these cells, they have to isolate the cells, they have to grow the cells in culture, and then you can do genetic testing. You can do the chromosomal analysis, the karyotypes. You can test for particular traits, particular syndromes and diseases that can happen in the family. But at the time that you can get this done, you are in the 18 to 20 weeks of pregnancy, which means by 21 weeks you can have a viable baby out of this. It takes about 2 weeks for these cells to be cultured. So you're not going to figure out if anything is wrong with the baby until you are in 22 or 23 weeks into the pregnancy. And now you get into those issues. How ethical is it to terminate a pregnancy in that stage? Very big issues that every person will address it in a different way. And it can be life-threatening, as in case of preeclampsia, it was definitely life-threatening. Coming back, amniotic fluid, by the time you can test those cells, it might be a bit too late for an ethical decision to be made. What are the advantages and disadvantages of amniocentesis and amniotic fluid testing? Okay, that's important to know. So you can get it by 18 weeks. By the time you get the results, it might be, a bit too late. So you go for another testing that can be done, this chorionic villus sampling, CVS. This also has advantages and disadvantages. So here you have the chorion, which is the part where the baby is attaching, right, to the uterus. And this test can be done at a much earlier stage than the amniocentesis. So here you have a fetus that is about 10 to 11 weeks old. So this is less than 3 months old, 3 months pregnancy. You go through the vagina, insert this catheter, get a little sample of this chorion cells, okay, and take them out. Usually when you get these samples, there you know that it's going to be still cells from the mother or cells from the fetus, but the amount of cells that you're going to get from here is a lot more than the amount of cells that you get out of the amniotic fluid. Okay. Sometimes you have to go for 2 or 3 extractions of fluid in order to make sure that they have cells from the fetus. Now in this you get the cells, and they grow a lot faster. Usually in about a week you can do the DNA testing and the karyotype, and you can figure out if anything is wrong with that baby. Now disadvantages of this type of method is that sometimes this procedure, the procedure itself, will cause a miscarriage. So if you're worried that the baby might have some problems, and then you go through this procedure, and then there you go, you have no more baby, cause a lot more miscarriage than the amniocentesis would. Another problem is that there is a borderline in which you can do this testing. If you do this earlier than 8 weeks of pregnancy, there is a high risk that the baby will develop some type of birth defects. Okay? Short limbs or absence of limbs, and some other types of birth defects. We don't know why really, because the baby should be by itself here, is only connected through the umbilical cord. Why is it that changing some of this, taking out some of these cells is going to affect the baby? Epigenetics, right? Something to do with the environment. It sucked up some cells that needed for some genes to be expressed, and especially at that early stage, right? This chorion is very, very important for the development of the baby. Now we go for the other testing. We have advantages and disadvantages. The faster culture is one of the advantages. Another advantage, of course, if you have the results back within a week, and then you have 12 weeks of pregnancy, and then you say, look, these chromosomes are really messed up, then at least you have some time to think about what to do. It's not like you have a viable baby at that stage. Okay, so the last part of chapter 6 is just talking about how we can compare genes between species, and you can basically see what would make people unique, and you compare to the DNAs or to the genes of other apes. So 2 genes that are very important. Actually, there's not a lot of difference between the genetic composition of the humans and the apes. I believe it's only about 2%. Less than 2%, right? Wow, isn't that scary? I mean, or I do believe the chimps are very smart. It's just that they cannot talk about it. And yeah, one of the genes that really make them not to talk about it is this FOXP2 gene that regulates human speech. So I was just wondering if you just make this genetically engineer the chimp with the FOXP2 gene if it will actually talk about it, you know. And another one is this microcephalin 1 gene. Well, actually, the way this gene was named is a bit the opposite of what it means. If the gene is not expressed, if there is a problem with the gene, you say you get microcephaly, which means a very, very small head. But then usually the genes are found out based on what kind of disease they cause, right? It's usually the type of negative finding out of things. So if the gene is not working, you say, oh, we got a problem. Doesn't mean you name the gene microcephalin. I would name it macrocephalin. So anyway, you know where the gene idea is coming from and what it's doing to the head. So this is just a little chart of comparison between overall identity of humans to chimps and humans to mice, which, you know, mouse is our favorite animal model for studies in the lab. There's not a lot of things in common between us and the rodents. But considering the chimp, you do have a lot of bases and DNAs and chromosomes in common.